Creating a brand new medicine from scratch is one of the biggest challenges in modern science. I mean, you're basically on a hunt for one perfect solution among countless, and I mean countless possibilities. So today, let's dive into this amazing world of pharmacophore modeling.
Okay, to really get this, imagine a biological target in our body, like a virus or a faulty protein, as an incredibly complex lock. To stop it, you need to design the one perfect key that fits. The problem? The number of potential molecular keys is in the billions, maybe even trillions. So where on earth do you even begin to search?
Yeah, this massive, seemingly impossible search is the central challenge that drug designers face every single day. It's the ultimate needle in a haystack problem. But what if you didn't have to search for the key itself? What if you could find the blueprint for the key instead?
And that, right there, brings us to the hero of our story, the pharmacophore. Now, it's not the drug molecule itself. Think of it as the secret design, the essential blueprint that tells us exactly how to build the perfect key.
Now, the official scientific definition from IUPAC, well, it sounds a bit dense, right? It talks about an ensemble of steric and electronic features. Really, that's just the precise scientific way of describing the specific 3D pattern a molecule needs to have to interact perfectly with its target. So let's just cut through the jargon. A pharmacophore is the absolute minimum set of features, arranged in a very precise 3D pattern that a molecule has to have to fit into that biological lock and turn it. It's the blueprint, with all the non-essential stuff stripped away. So what kind of features are we talking about? Well, they're fundamental chemical properties that act like the specific ridges, grooves, and notches on our master key. We're talking about areas that repel water, special chemical rings, and points that can form these crucial hydrogen bonds, all positioned perfectly in 3D space.
And this highlights a really crucial distinction. A drug molecule, the actual key, is made of two parts. You've got the pharmacophore, which is the busyness end that actually does the work by fitting into the lock. Then you have the rest of the structure, which you can think of as the handle of the key. It's the backbone holding those essential features in just the right place and, helping the body process the drug.
So, the whole point is, if we can figure out the blueprint, we can design the key. But how do scientists actually find this blueprint? Well, it turns out there are two main paths, two different detective methods they can use.
This really gets to the core question for computational chemists. How do you reverse engineer this thing? Do you study the lock itself, or do you analyze a whole collection of keys that you already know work?
And this lays out the two main strategies perfectly. On one hand, you have structure-based modeling, where you literally analyze the lock, the biological target protein. On the other hand, you have ligand-based modeling, where you analyze a set of existing keys, molecules you already know are active, to find their common design.
All right, let's look at the first method. This is the most direct approach. If you have a high-resolution 3D picture of your lock, you can design a key specifically for it. The process here is super logical. Scientists map out the binding pocket of the target protein, and they identify all the spots where a molecule could form a bond or interact. Then they just create a pharmacophore model that is a perfect mirror image of these interaction points. It's pure, elegant, structure-based design.
But, and this is a huge but, for a lot of important biological targets, we simply don't have a clear 3D structure. The lock is a black box. So what do you do then? Well, that's when you have to switch to the other detective method. If you can't see the lock, you study the keys that work. There's a really powerful algorithm for this. First, you find all the common 3D features across a set of molecules that are highly active. Then, and this is the really clever part, you subtract any features that are also present in molecules that are inactive. This isolates the exact pattern that's responsible for success. Finally, you optimize that pattern into a single, predictive pharmacophore model.
Okay, so now we have our blueprint. We've got our pharmacophore model, created by one of those two methods. What's next? This is where the magic really happens. We're about to put this digital locksmith to work on a massive scale.
We've done the hard detective work. We have the perfect design specifications. Now it's time to go from that blueprint to finding actual candidate molecules.
Scientists have access to these enormous virtual databases. We're talking millions, sometimes tens of millions of different chemical compounds, all mapped out in 3D. Trying to search this manually would be completely impossible. It is our entire vault of potential keys.
And this is the payoff. The Pharmacophore blueprint isn't a physical thing. It's a search query. It's a highly specific 3D digital filter that you can apply to that entire massive database in an instant.
Just think about the power of this. You start with millions of candidates. You apply your Pharmacophore filter, and only the molecules whose 3D features perfectly align with your blueprint make it through. Suddenly, that impossible vault of billions of keys is reduced to a manageable shortlist of a few hundred, or maybe even a few dozen, promising candidates that can actually be synthesized and tested in a real lab.
Pharmacophore modeling has completely revolutionized the early stages of drug discovery. So let's wrap up by looking at why this is so important and where this amazing technology is heading.
The impact is just immense. This whole approach turns a slow, expensive trial and error process into a fast, focused, and intelligent hunt. It saves an incredible amount of time and money, and it allows scientists to focus their precious resources on the molecules that are most likely to work. It's truly a cornerstone of modern drug design.
And the innovation definitely doesn't stop here. This is a synergistic approach. Scientists are now layering this technique with other computational tools, like molecular docking and artificial intelligence, to refine the search even further, making these blueprints smarter and more predictive than ever.
Which leaves us with a final, fascinating thought. We are getting so much better at designing these master keys for diseases. As these molecular blueprints become more sophisticated and powerful, how long will it be until we can design a key for any lock, turning diseases we currently consider incurable into manageable conditions of the past? The hunt is on.